This video is the final one in the unit on DNA and proteins, and the topic is Biotechnology Part 2, looking at gene editing. So when we look at traditional biotechnology, over here we've got PCR, genetic analysis, we've got transfer of genes into cells to change gene expression to express new proteins taken from existing organisms into new organisms. And this is really covered in the first couple of dot points of the student learning outcomes there. What this video will look at is the second to last and the last dot point there around what are some of the new technologies that are not just using existing proteins in new organisms, but actually altering the properties of existing proteins so that have different characteristics. And this involves the CRISPR-Cas9 system and protein design. So CRISPR-Cas9 is really quite new. It's only really been used widely in the last decade or so. And it's a really fascinating system, originally derived from study into what's going on in existing cells, but now being exploited as with all biotechnology. Like I said, existing biotechnology looks at taking genes that make proteins from one cell, from one organism, and putting those into another, adding those on top so that there's a new gene in that organism. But they don't, those technology can't really address changes or functions to existing genes within the host organism. And that's where CRISPR-Cas9 comes in. So CRISPR-Cas9 is actually a two-part system. The CRISPR refers to this RNA, that is a guide RNA, and that has a specific sequence, a little bit like an RNA probe, that will find a target sequence on a stretch of double-stranded DNA. The Cas9 is an enzyme, that's a protein that will cut that double-stranded DNA. So the two parts, the RNA finds the particular part of the protein, and then particular part of the DNA, and then the protein cuts that DNA. And the cell will normally then repair those double-strand cuts. But what can be done with the system to modify it slightly is that during that cut, there's an insertion of DNA, or small bits of DNA can be cut out. And so depending on how you set up the system, you're able to remove one nucleotide, or add one nucleotide or a few nucleotides, or switch one, like an A for a G or something like that. And so it's remarkably powerful to be able to edit that DNA. The other really powerful part of it is, whereas restriction enzymes will cut DNA, they will only cut at a certain type of sequence that they find, and every restriction enzyme has a different sequence it'll cut at, and you can't cut at any sequence that you want. Cas9 will cut anywhere as long as it's directed to by the guide RNA. And the cool thing is, the guide RNA, you can synthesize that, make that any sequence you want. And so CRISPR-Cas9 will find your particular target sequence, whatever it is you've set up, by changing the guide RNA, and it will edit that part of your double-stranded DNA. So remarkably powerful to be able to edit existing genes in place, rather than just parachuting in a whole extra gene, which is the sort of brute force approach that old biotechnology uses. Along with that, then, we've got this side of things, which is protein design. So when we are editing genes or changing things around, that's going to change the protein, obviously, and that's the reason that CRISPR-Cas9 system is used to change the gene. You're going to change the properties of the protein. Because, of course, DNA codes for RNA, codes for an amino acid sequence that will lead to the protein structure. You change the DNA, you change the structure. So if we're going to manipulate proteins and change their properties, we're going to actually, what we're doing is manipulating DNA to do that. And so why might you do that? Well, you might want to change the property of a protein that's an antigen that's causing an immune response in a vaccine. By changing the properties of that protein, you might get a bigger immune response. If it's an antibiotic that's killing bacteria, by changing the properties of that protein, you might increase the toxicity of the antibiotic. And in the last case there, which was related to the 2008 Nobel Prize for Chemistry, there's a green fluorescent protein that's found in jellyfish, and scientists developed the use of this as a marker for where proteins are in cells, because whatever you attach to that protein is going to now have green fluorescent tag on it. But that was limited to one color. By doing this sort of rational design directed evolution, you could change the properties of that protein so it actually fluoresces in different colors. So there's now yellow, red, blue variants of that. So how do you do that? Well, there's two approaches. One's sort of the more old-fashioned, one's a bit more modern. The old-fashioned approach is a directed evolution. So you do random mutagenesis. You randomly create mutations in the gene of interest and stick all those different variations into a plasmid. Transform, which means to put those into bacteria, express the protein, and then look at the proteins that are made for their properties and select the ones that have the best outcome. So for instance, you could do mutagenesis on catalase, look at all those different versions of the catalase and see which one breaks down hydrogen peroxide fastest in order to improve the properties of that. If you're looking at doing that on DNA polymerase for PCR, you could mutate it and then see which one still works at a very high temperature like 98 degrees Celsius and look at which ones are still working at that temperature. On the other hand, as we've learned more and more about protein structure and how amino acid sequence leads to different protein structures, we can start to actually do CAD, computer-aided design on proteins. So you have an existing protein, you think it would work a little bit better if we could open out the active site or twist it around a little bit. And so in order to change the shape of the active site, these are the amino acids that we want to change their properties slightly. And so we can work out from those amino acids what the DNA sequence needs to be in order to do that. And so using CAD, we can do a site-directed mutagenesis. So mutate or change that particular base. That's going to lead to that change in the amino acid. CRISPR might be one way of doing that. Put that into bacteria, express that, purify that protein, and there you've got your new version of the protein. So either randomly doing it and selecting which one's best, or rational design about this is the change I want to make and making that in the DNA. And then you can go through that process, either process, again in lots of cycles until you get the improved protein characteristics that you want. So biotechnology has come a long way since the 20th century. 21st century biotechnology is now very targeted and specifically able to change particular sites of DNA in existing cells, change that DNA around, making the proteins different. There's still a long way to go, obviously, but the technology is improving all the time. That's the end of the DNA and proteins topic. Hope you learned a lot during that, and it's time to start preparing for the test.